Allergies are violent immune responses triggered by innocuous proteins, referred to as allergens. Although some allergens, you may argue, are not totally harmless. Bee stings, for example, can be very dangerous, but not because of the actual venom, but rather due to an allergic overreaction. Allergy to bee stings can lead to dramatic swelling of the throat and tongue, shortness of breath, vomiting, and a sudden drop in blood pressure. This response is known as anaphylaxis, from the Greek, against protection, and can be very serious. Fortunately, these reactions can be reversed with a shot of epinephrine. Epinephrine reduces the permeability of blood vessels and so stops the swelling, relaxes constricted airways, and stimulates the heart, which together restores our blood pressure. So we see overreaction can be life-threatening. In this unit, we will see how the other extreme, the inability to raise a functional immune response, is equally life-threatening. As it turns out, there are a number of reasons why we might experience immunodeficiency. Surprisingly, the most common cause of immunodeficiency is malnutrition. In poverty-stricken countries, in times of famine, we often hear of people dying of starvation. Have you ever considered what that actually means? People who are severely undernourished lack energy, strength, and suffer from failing metabolism. Death is often due to the inability of starving people to raise a functional immune response and combat infectious disease. Fat cells produce a hormone called leptin, which among other functions is very important in the development of T cells. As undernourished people lose their fat storages, the fat cells stop secreting leptin. Our T cell responses drop as well, and we become particularly susceptible to infection, compromised and unable to protect ourselves. Clearly, as we now understand, a vibrant and robust immune system demands healthy and functional T cells, and so eating well is important and ensures a healthy immune system. A second cause of immunodeficiency is congenital birth defects. Some genetic diseases impair the production of lymphocytes. SCID, severe combined immunodeficiency, is such a genetic birth defect, in which central mechanisms crucial for the development of both TCR and BCR are lacking. Thus, kids born with these mutations have no functional B or T cells. Such babies have to be isolated and protected in germ-free, sterile environments, plastic bubbles. David Vetter was the most famous bubble baby who survived 12 years in his bubble until ultimately succumbed to a viral infection that led to lymphoma in 1984. Fortunately, with modern technology, diagnosis of SCID is now possible at birth, and bone marrow transplants can restore the immune system and actually cure SCID babies for good. The third cause of immunodeficiency is certain viral infections. In June 1981, the CDC, the Centers for Disease Control, reported 5 cases of a rare fungal pneumonia called Pneumocystis carinii pneumonia, or simply PCP. All 5 patients were young homosexual men living in California. By December of 1984, over 125,000 cases of PCP were reported in the United States and shown to be caused by a recently emerging virus, HIV. HIV, the human immunodeficiency virus, directly attacks T cells and so causes AIDS, acquired immunodeficiency syndrome. HIV is a positive strand RNA virus, about 120 nanometers in diameter. Its genome is about 9,800 bases long, coding for 19 proteins. Each viral particle contains 2 identical copies of the RNA packaged in the capsid. The viral spike protein is made of 2 subunits, GP120, which is linked to the membrane-bound GP41. HIV is a retrovirus whose replication is the most complicated virus lifestyle. Here, HIV is approaching a susceptible T cell.
Its viral spike protein, GP120, binds to CD4, the cell surface protein that the virus uses as its receptor. This binding triggers a series of events where GP120 shifts a bit, and as CD4 moves aside, the viral spike can then interact and bind with other cell surface proteins that ultimately lead to membrane fusion to release the capsid into the cytoplasm. The capsid breaks down, and the unique process of reverse transcription begins. The viral enzyme, reverse transcriptase, transcribes a first strand of DNA off the viral RNA template. Next, a second complementary DNA strand is produced. This double-stranded DNA copy of the viral genome then proceeds and enters the nucleus. The cellular chromosome is nicked, and the viral DNA integrates into the cell's chromosome. At some point, the cell's RNA polymerase, the enzyme that naturally produces messenger RNA, begins to produce HIV RNA genomes that exit into the cytoplasm. As you recall, the HIV RNA is a positive strand. Thus, ribosomes can begin to produce all the viral proteins, the capsid proteins, the viral enzymes, and new spike proteins that appear on the cell membrane. The viral RNA is then packaged into capsids that bud out of the cell, enveloped in cellular membrane containing viral spikes. These new viruses can continue the cycle to infect more T cells.
Ultimately, there are too few T cells to mediate effective immunity. And like in the case of malnutrition, our body is simply unable to fend off disease. Our weakened immune system provides opportunity for pathogens to overwhelm us. Hence, people suffering from AIDS are said to succumb to opportunistic diseases. I've asked Professor Robert Gallo to explain how by inventing a blood test, HIV was proved to be the cause of AIDS.